Hello, biologists. Today we're going to talk about meiosis. So the first thing we want to do is define the goals and uses of meiosis. Meiosis is going to create 4 non-identical daughter cells. That means that you'll have 4 genetically different cells by the time you're done with meiosis. Meiosis is used to produce sex cells. Specifically, we call those gametes. In humans, those gametes are called sperm and egg. In other organisms, they're called different things. Like in fungi, they're called spores. And in plants, it's called pollen and ovules. But overall, these are the cells that are going to be used for sexual reproduction. Now, if an organism is diploid, those sperm and egg will be haploid. The reason that they have to be haploid is because you get half of your genetic information from each of your parents. So we have 46 chromosomes. We get 23 from mom, 23 from dad. Each of those, the sperm and the egg, are haploid, so that when they fuse in fertilization, you get a diploid zygote. So haploid means one set of chromosomes. Here we have an overview of the process of meiosis. In meiosis, we are going to take our chromosomes. I have one from mom and one from dad. And then I have another one from mom and one from dad. So I am looking at 4 different chromosomes. 2 of them are paternal and 2 of them are maternal. During interphase, I'm going to duplicate the DNA just during S phase like I did in mitosis. Now I have 4 chromosomes, each is duplicated, and they are sister chromatids, one on each side, held together at the centromere using those cohesion proteins. So you have duplicated your chromosomes, they are visible, and then you do 2 rounds of meiosis. In each round of meiosis, you'll go through the steps of prophase, metaphase, anaphase, and telophase, but it's a little bit different in meiosis 1 than it is in meiosis 2. The really interesting things happen in meiosis 1. The first thing that happens in meiosis 1 is you go from being a diploid organism to a haploid organism. And I'm saying organism, but I meant cell. A diploid cell to a haploid cell. So here I have a set of this chromosome from my mother and my father. And now in my daughter cells, I have one of the little chromosomes from mom and one of them from dad. So I'm no longer 2 sets of DNA. I have 1 set of DNA. So that occurs in meiosis 1. Another thing to note, during the process of prophase, I'm going to have crossing over occur. Crossing over is where pieces of the DNA swap with their homologous pairs, and we'll talk about that in 1 second. And what you're left with is genetic recombination. So the chromosomes that I started with are no longer identical. And then in meiosis 2, you're going to go through prometaphase, metaphase, and anaphase and telophase again, and you are doing a second round of division so that when you are done, you end up with 4 daughter cells. Each is genetically different, and these cells are haploid. Here's the process of meiosis. In interphase, your DNA is going to duplicate, the cell is going to get larger, the cellular components are going to duplicate, your centrioles are going to appear, the plasma membrane will be intact, but the nuclear membrane will start to break down. In prophase 1, your chromosomes are going to condense. They're going to attach to your spindle apparatus. The nuclear envelope, which is being shown in white right here, will completely disintegrate, and you will start to move your chromosomes to the metaphase plate. In metaphase 1, your chromosomes line up at the metaphase plate, but look, they're not lined up as single chromosomes. Now they're lined up in homologous pairs. A homologous pair means that the chromosome sets that you got from your parents line up together. So we have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3 because we are diploid. We got 1 from mom, 1 from dad. The maternal and paternal chromosomes line up in a set. So say this is chromosome 1. I have a copy of chromosome 1 on top of another copy of chromosome 1. These are called homologous pairs, homo being same. Homologous is a word means evolutionary origin. So these chromosomes are the same size and shape, and they carry the same type of alleles, but it's your sets together. When we did this in mitosis, it was just one chromosome lined up on the metaphase plate. Now we have two chromosomes lined up on the metaphase plate. If we look at this green chromosome, it is two sister chromatids attached at the centromere, and it's being lined up with the pair that is similar to it from your mom or your dad. So these are homologous pairs. I have two chromosomes right here, each chromosome made of a set of sister chromatids. So this is different. During prophase, each of these chromosomes that sets up with its homologous pair is going to smush really close with its neighbor, and it's going to end up swapping genetic information with each other. That's when crossing over occurs. So crossing over is setting up here. It's still occurring here. And then when I pull my chromosomes apart during anaphase, I am separating my homologous pairs, not my sister chromatids. So from this point on, I no longer have homologous pairs. I will do telophase and cytokinesis like normal. So my nuclear envelope will reform, the chromosomes will decondense, the aster will disappear. And now I have two daughter cells. Both are haploid, but they both have duplicated chromosomes. So I need to divide them again to separate my duplicated chromosomes. So each of these two daughter cells will go through another round of division. We call that meiosis 2. In meiosis 2, your chromosomes will condense, the nuclear envelope will disappear, the spindle apparatus will attach to the chromosomes at the kinetochore, and you will line up your sister chromatids in the middle of the metaphase plate. Now, during anaphase, when those adhesion proteins disintegrate, I can separate my sister chromatids, and they will pull to opposite poles of the cell, so that when I do telophase 2, my chromosomes each have one copy of a chromosome. So meiosis 2, very similar to mitosis. Meiosis 1, not similar to mitosis because crossing over occurs and my homologous pairs are separated instead of my sister chromatids. When I am done, I will have 4 cells that are genetically different from each other, and those will turn into sperm or egg. Let's talk a little bit about crossing over. I've got 2 visuals for you. Let's look at this bottom one first. Here I have homologous chromosomes. These are my homologous pairs. This is chromosome number 5 from my mom. I've duplicated chromosome number 5 during interphase, so I have 2 copies of chromosome 5 held together at the centromere using the proteins in the kinetochore. So this is 1 copy of chromosome 5. This copy came from my mom. This is going to be chromosome 5 from my dad. Chromosome 5 from my dad is also duplicated and held together at the centromere. These are homologous pairs because they're both chromosome 5, but one is maternal and one is paternal. During crossing over, my homologous pairs get really close together, and we call them a tetrad, tetra being 4. So the tetrad gets really close. They kind of like adhere and smush and mesh together so that when they pull apart later, they've end up swapping DNA. So this sister chromatid is fully my mother's, but this sister chromatid is mostly mom's DNA, except it has a little bit of paternal DNA that it swapped with the other chromosome during crossing over. This allows us to recombine our genes and create genetic recombination. Here's another visual. During prophase and prometaphase, my homologous pairs, this being one chromosome, this being another chromosome, I have 2 of them because I'm a diploid organism. They're going to get really close together. They're going to end up swapping pieces of DNA so that when they pull apart during anaphase, they are no longer genetically identical sister chromatids. In mitosis, the 2 sister chromatids stayed identical. Crossing over makes my sister chromatids no longer identical. Then in prophase and metaphase 2, I'm going to line up my single chromosomes on the metaphase plate, and I will make the cohesion proteins between them disintegrate so that in anaphase 2, I separate my sister chromatids. So in anaphase 1, you're separating your homologous pairs. You're going from diploid to haploid. In anaphase 2, you're separating your sister chromatids so that each daughter cell only has one copy of the DNA. I like this visual crossing over because it shows all the proteins that are meshing between them. The proteins between your homologous pairs are called the synaptonemal complex, synapse being where two cells touch. So here you can see your centromere is the pushed in part of the cell. The kinetochore are the proteins that are holding your two sister chromatids together. So I have this red chromosome is chromosome 1, and this is an exact duplicate of chromosome 1. I have two copies of chromosome 1. This blue one is also chromosome 1, but it's from my other parent. So because I'm diploid, I have two sets of chromosome 1. That's where homologous pairs come from. And the DNA of those homologous pairs was duplicated during interphase. So I have two chromosomes, two copies of each. If we look closely at the tetrad when it's formed, we can see the synaptonemal complex between them. And that's what allows pieces of DNA to change location. So it's literally binding and recombining as it is sitting there together in prophase. Another thing that gives us genetic recombination is independent assortment. Now, sometimes people say random orientation. It's slightly different, and I want to clarify the difference between each other. This is going to be a visual of independent assortment. So here's possibility number 1. When my homologous pairs are lined up on the metaphase plate during meiosis 1, I have all my maternal pairs on the left and all my paternal pairs on the right. When I divide this cell, I'm going to have these maternal duplicated chromosomes in one daughter cell, and these paternal duplicated chromosomes in another daughter cell. During meiosis 2, when I separate my sister chromatids, I will now be left with 4 daughter cells. 2 of them contain maternal DNA, and 2 of them contain paternal DNA. Now, that doesn't have to be the case. Because of independent assortment, whichever one is on the right or the left is totally independent, right? So if this one's on the left, it doesn't change whether or not the chromosome behind it aligns in the same way. I could very equally have this possibility where I have chromosome 1 that's maternal on the left and chromosome 2 that's paternal on the left. And now when I do my first round of meiosis, I no longer have just maternal DNA in my daughter cell, and my outcome from meiosis 2 is different. So we get all this different recombination based on who lines up on the left or the right, and that's totally random. And this is showing, just 4 chromosomes. We have 46. So think about all the variables of who goes into what cell based on which is on the left and which is on the right. So there's a lot of variation that you can get from independent assortment. Random orientation is a little bit different. Random orientation is the idea that the alleles on each chromosome are independently assorted from each other. What does that mean? That means if I go back to this picture here, this picture here, whether or not I got the big G or the little g on one of my chromosomes is independent of the genes below it. So I don't have to have all my types of alleles that are dominant on one chromosome or recessive on the other chromosome. So the inheritance of one gene is not influenced by the inheritance of another. A nice little table for you here to get the definition between them. I'm not going to ask you to clarify the definition between them, but I do think it's important to know that they are different. All right, let's check our vocab. A homologous chromosome, a tetrad, and a sister chromatid. Ask yourself, how are those different? And then ask yourself, how is gene different than allele? How is genotype different than phenotype? And how is heterozygous different than homozygous? All right, I'm going to answer them. Sister chromatids are identical copies of a chromosome that are held together at the centromere. Here we see 2 sister chromatids held together at the centromere. Sister chromatids are identical. A homologous chromosome is the chromosome number that you inherited from both your parents. We have two sets of them. So our homologous chromosomes are maternal and paternal. Now I'm saying we're diploid. Things like strawberries are tetraploid or octoploid. So they would have gotten 4 copies of chromosome 1 from their mother and 4 copies of chromosome 2 from their father. So it's not always 1, it's whatever number of copies you have. So homologous chromosomes are your maternal and paternal DNA. It is chromosomes that are numbered the same, they're the same size, and they contain the same genes. So these are homologous chromosomes because they both are the same size and they contain the genes G and R in the same locations. A tetrad is when your homologous pairs line up really closely during crossing over and allow that synaptonemal complex to form, and crossing over can swap DNA between your homologous chromosomes. A gene is a piece of DNA that codes for a protein. An allele is a different version of that gene. So here we see a gene for gene G. You can have the dominant allele or the recessive allele. If you have 2 of the same type of the allele, you are homozygous, homo being same. So this chromosome is 2 copies of big G. This is 2 copies of little g. If you're heterozygous, you have 1 of each, hetero being different. Over here, we see 1 big G, 1 little g. So those would be heterozygous. I want to compare mitosis and meiosis. So in mitosis, you start with 46 chromosomes, you produce 2 daughter cells, and they each have 46 chromosomes. Mitosis takes diploid cells and produces diploid cells. Mitosis produces identical daughter cells, and mitosis is used for growth, repair, development. Mitosis creates somatic cells only. Meiosis, we're going to start with 46 chromosomes, and we're going to end with 4 daughters with 23 chromosomes. So we've gone from a diploid cell to a haploid cell. We're doing 2 rounds of division because we need double the amount of cells. We are not making body cells. We're making only sperm and egg. And these cells are genetically different from each other. So there's genetic recombination. The daughter cells are different from each other, and they're different from the mother cell. I also have this nice Venn diagram for you to look through and make sure that you feel comfortable with the differences between meiosis and mitosis. It's also nice to look at the 2 processes together. So on the left, we have mitosis, and on the right, we have meiosis. Both of the parent cells that start this are diploid. We denote diploid by 2N. N is the number of chromosomes you have. We have 2 sets of chromosomes, so 2N. If I was tetraploid, it would be 4N. If I was octoploid, it would be 8N. In this example, we're showing 4 chromosomes. So this diploid organism has 4 chromosomes. When you do mitosis, your DNA is duplicated. Your sister chromatids line up on the metaphase plate. And when you separate it, you have 2 daughter cells, each with 4 chromosomes, and they are diploid. They're identical to each other, and they're identical to the mother cell that we started with. In meiosis, we're going to do 2 rounds of division instead of 1. We're going to have crossing over occur in prophase. So that's going to add to genetic recombination, making our daughter cells genetically different. In the first round of division, homologous pairs line up on the metaphase plate. When those pull apart, we can see that crossing over occurred. At this point, we've gone from diploid to haploid, but we still have double the amount of DNA. And so we need to do another round of division. In the next round of division, your sister chromatids will line up on the metaphase plate, and they will be separated so that when you are done, you have 4 daughter cells. Each is genetically different from each other, and each is genetically different from the mother cell. And these are haploid, where we started with a diploid organism. So now there's only 2 chromosomes in here, where there were 4 in the beginning cell. I've got a bunch of videos for you to watch on Blackboard. The first are 2 visuals of mitosis happening in real time, which is so neat. I really think it's worth checking out. Then I have a mitosis animation, followed by a cell cycle and mitosis review. So that's for your mitosis. I have 2 informational videos about Henrietta Lacks. And if you haven't heard about Henrietta Lacks, I'm really excited for you to learn about her because she did such an important thing for modern medicine completely without her consent, and her family had to struggle to even get her recognized, and she deserves to be recognized. Henrietta Lacks is an American hero, so we should all know her name. So check out those 2 videos. And then we have 2 videos of meiosis going through the processes. So it's a lot to watch this week, but there's a lot of technical steps and processes to get down. So as you go through, please let me know if you have any questions. I'm happy to help. Email me anytime, and I will talk to you soon. Bye.